to kickoff weekend for Polar DinoFest, a 10-week exploration into the world of polar dinosaurs. My name is Randy Ermis. I'm the Chief Curator and Curator of Paleontology here at the Natural History Museum of Utah, and I'm zooming to you right from the paleontology collections. Before we get started, I want to remind everyone to kick off Polar DinoFest. We're hosting a total of 6 live stream panels this weekend with a total of 12 guest paleontologists. This is our last live stream panel for today, but we'll start again tomorrow at 11 a.m. Mountain Time with further panels at 2 p.m. and 5 p.m. tomorrow. These live conversations kick off 10 weeks of polar dinosaur exploration as we release a new video from one of our guest paleontologists each Friday at 10 a.m. Mountain Time between now and April 2nd. You can see the complete schedule of our live conversations and the video release dates on our website at nhmu.utah.edu/DinoFest. And we encourage everyone to submit their questions via the website, Facebook, or YouTube. Now let's get started. I'm really excited to be joined by one of our keynote speakers and fellow paleontologist, Dr. Holly Woodward Ballard, Associate Professor of Anatomy and Paleontology at Oklahoma State University Center for Health Sciences. Welcome, Holly.
Thanks, I'm really excited to be here.
Well, thank you for joining us this evening. Before we get started with the questions, could you tell us a little bit about how you got involved in paleontology in the first place?
Sure, I was one of those that loved dinosaurs since I was a little kid. I can't remember a time when I didn't want to be a paleontologist. When I was little, I would dig in my yard in the dirt trying to find dinosaur fossils in the Piedmont of North Carolina. So that was kind of fruitless, but and from there, I was always fascinated with microscopes as well, because I really like the way you can look at things on a high resolution scale and see even more information. So it was really exciting for me to learn about bone histology, where I could marry my two interests together with dinosaurs and with microscopes. And so I didn't really know what I wanted to study as far as dinosaurs or paleontology went. I knew I wanted to focus on dinosaurs, but as far as the field, I wasn't sure. But then when I found out about histology, I was like, that's it, that's for me. And so it's been smooth sailing ever since, as far as, I know where I am, I know this is what I'm good at.
Yeah, so many of our viewers might not have heard of histology before. Could you explain a bit more about what kind of paleontology that is?
Yeah, sure. So histology is the study of tissue microstructure, and I study bone histology because in the fossil record, the most frequently kinds of preserved fossils are bone. Soft tissue rarely preserves. So if you are to look into, or if you were to take a tiny little slice of a bone from an animal today, put it under a microscope, you would see bone cells, you would see blood vessels, you would see the bone fiber, and all of this can tell you information about how an animal was growing. And because, all vertebrates have bones, there's not a difference in composition between our bones and that of a dinosaur. So the same structures you see in modern animal bone, you can see fossilized dinosaur bone has the same thing, which is always really exciting for me to think that, this bone that I'm holding in my hands, even the microscopic, cellular structures are preserved. And so by doing that, you can look at dinosaur bone under a microscope and figure out how fast it was growing. Bone has tree rings. Well, they are like tree rings, but they're not from a tree. So these are rings that happen every year when an animal stops growing. And you can count them and then figure out how old that animal was when it died. So they look a lot like tree rings. So that's the reference there. But it's a really cool field or subfield. And I'm super excited to be part of it.
Yeah, so that gets to one thing about fossilization. I think, we often hear in school that fossils are bones turned to stone or replaced by minerals. But it sounds like what you're telling me is in a lot of cases, the original tissue is still there.
Yeah, so the really interesting thing about bone is that it has a soft tissue component and a mineral component, and which gives it flexibility but strength. So the mineral component is what gets preserved in the fossil record because it's it's hard. They're basically crystals. The soft component is what decomposes away, but they parallel or mirror each other. So whatever the soft tissue is doing as far as its organization, the mineral is basically lining up in the same direction. So even though the soft tissue in a bone is typically not preserved, we know what the soft tissue was doing because the bone fibers are doing the same thing. So even though we don't have that soft tissue to look at the actual bone cells or the actual blood vessels, we can see where they used to be, and we can see how the bone was being formed.
That is so cool. When you sort of talked about all these different types of ways we can use histology to understand dinosaur growth and dinosaur biology, some of the work that you've done has involved dinosaurs from the Arctic.
Yeah.
Could you say a little bit more about how you got involved in working with Arctic dinosaurs?
Sure, yeah. So by the time I started getting interested in the Arctic dinosaurs or the Antarctic dinosaurs, I was already a graduate student studying bone histology, so I knew that's where I wanted to focus. And I happened to see a presentation by Dr. Tom Rich, who was visiting from Melbourne, Victoria, in southern Australia, but he was up in Montana to look at the fossil collections there. And he gave a presentation on the dinosaurs of Australia. And what is really cool to think about is the southern part of Australia used to be connected to Antarctica. And through, millions and millions of years of drift, it's drifted further towards the equator. But during the Cretaceous, when dinosaurs were around, this part of Australia, southern Australia, was within the Antarctic Circle. So the dinosaurs that were living there were experiencing different conditions that other dinosaurs living around the world wouldn't have had to deal with, which includes colder temperatures and, longer nights, complete darkness for several months, as well as months of complete brightness. And so I was curious into how these dinosaurs actually were adapting to this environment. And so I asked Dr. Rich if I could visit his museum in Australia and histologically sample some of his specimens, because what I wanted to know is, like, from babies to adults, what's going on in their bone structure and what can I learn about how they're growing in such an environment. So that's basically it. He said, yeah, come on down. And who wouldn't want a chance to go to Australia?
That's awesome. And that's a perfect lead in to one of our first questions from Amy, all the way from Giovano, Italy, who wants to know what particular adaptations did these polar dinosaurs have for living in the North or South Pole in this case?
Oh, that's a great question. So the polar dinosaurs that I looked at are, they would have been about the size of a turkey, or for Australians, a wallaby. So they weren't able to migrate long distances. So whatever conditions they were experiencing, they were stuck there. And so skeletally, looking at the bone tissue histology, we could see that these little guys were growing pretty much the same way as their relatives that were living across the globe. So they weren't doing anything differently physiologically. They were still growing at pretty much the same rate. They were staying small, but that's also, that was one of the really cool conclusions, I thought, of the study that I did, because it shows that dinosaurs, even these little guys, were already sort of pre-adapted to invade those niches that might be challenging for other types of animals, just because of their ancestry and the physiology that they inherited. I would assume physically that they probably had some adaptations that we just can't see, probably some sort of coating, feathers or hair or something. And maybe if they were burrowers, maybe they had larger eyes. So. Yeah.
Wow, it's incredible how much you can get out of just a single slice of bone. That's really amazing, and really has opened up so much new research into dinosaur lives.
Yep.
And we have a related question also about histology from Colin on YouTube, who's wondering whether histology can help distinguish between different species, either like recognize that you found a new species, or whether two specimens are the same species or not, things like that.
Oh yeah, definitely. So that's a great question. And the answer is yes, histology can help you do that. It was first actually done back in the 30s. So there was some debate on whether these two different duck-billed dinosaur hadrosaurs were the same species or whether they were different species. And by looking at the histology, you could tell the maturity level, like was one just older than the other, or were they the same level of, same age, but they're doing different types of growth strategies. So just by looking at the histology, in comparing the individuals, if they look similar, then they could be the same species. If they look completely different, they could be two different species. For example, with the work I did with the polar dinosaurs, I was studying a sample that was collected over a period of, stratigraphically 12 million years. And in this case, the histology was all very similar, but I would expect that over that 12 million years, I'm looking at different taxa. They just were growing the same way. So sometimes histology can't help in that respect, but a lot of times, especially in bigger animals that have a different type of growth trajectory, it can be really helpful.
Do we ever have the case where like we have dinosaur fossils from different places and they seem to be the same species, but they show different types of growth?
So they're from the same place, but they're growing differently?
From different places, but the same species. And, maybe one lives in a cold place and is growing differently. Yeah.
So this sort of jumps into the work that you might hear about from Pat Druckenmiller, but in the North Slope of Alaska, there are some ceratopsid dinosaurs, centrosaurs up there, and the histology was done on those individuals, and they were growing, they were pausing their growth very definitely every year, whereas the centrosaurs that you find further south didn't have such dramatic pauses in growth. So if you do the tree ring comparison, trees that are undergoing a really stressful winter will usually have, a thicker growth line there than in good years. And that's sort of what they were seeing with the ceratopsids in the North Slope, is that their growth rings were really pronounced compared to the ones further south.
Oh, that's super cool. And if you want an example of a centrosaur, I've got one right behind me. This is Diabloceratops from Utah. So you mentioned this pause between year, like in season, and also a little bit earlier, you talked about these growth rings that are kind of like tree rings, and it was Kim on YouTube was really interested in this and wanted to know more about how you estimate the age of a dinosaur using histology.
Yep, that's a great question. Everything that we do with histology, and pretty much everything we do to understand how dinosaurs lived, we have to ground in the world around us. So we look at animals that are living today and observe them to make inferences about what was happening in the past. So in order to understand what in the world these rings actually mean, we have to go and look at the histology of modern animals and figure out what those rings are telling us in modern animals. And so this is what scientists have done. They've actually tracked the rings and when they're actually formed in the bone, and they correspond to an annual occurrence. So every year, vertebrate animals will pause their growth for some amount of time. It leaves a ring in the bone, and then they resume growth until the next year. So because we know this is annual in modern animals, then we assume it was annual in dinosaurs as well. So we can count those rings and get an estimate of age at death.
Oh, that is so awesome. And I just want to remind people that you can continue to submit your great questions via the website, Facebook, or YouTube. So keep those questions coming. So something that maybe you mentioned briefly, but you sort of talked about is, if I understand correctly, you're actually cutting into these precious dinosaur bones.
Yep.
So how do you convince a museum curator to do this?
It's gotten actually easier over the years, and I'm so glad to say that, but I actually have some props because
Awesome.
I was wondering if someone was going to be concerned that I was actually cutting a bone. So this is a Maiasaura, a duck-billed dinosaur humerus. And so one of my graduate students is trying to figure out how the forearm of Maiasaura was growing. So he's done some histology on this, and The really cool thing is when you remove a piece of bone, you can make a mold and cast of that piece, which replicates the original in every way, every detail, and then you can reinsert that piece back into the bone. More recently, we have even more high-tech options like 3D scanning, which digitizes the specimen and the piece that you can take out, so you have a complete digital record of the bone. Some museums ask that we paint the cast and then reinsert it. So in this bone, this middle piece right here is the cast piece. So you can see how it looks really nice, just continuous. The only reason you could suspect it was a replica is just the two little cut lines there, but otherwise it fits back into the bone perfectly. So that's my first pitch for why you should let me do this. And the other reason is that we would have no idea about these dinosaurs, how old they were, sometimes if they were male or female, their growth rates, their growth curves, if we didn't look inside the bone. And I've been really lucky in getting to do this. Before I went to Australia, only one polar dinosaur from Australia had ever been histologically sectioned before. And I increased that sample to 12. So that doesn't sound like much, but for paleontology, that's a nice sample. Yeah. And then more recently, I've been looking at T-Rex. So I do have a prop here of, this is a slide of a juvenile T-Rex, the tibia. All the brown is bone. And it started out as a block. So I take the bone, I embed it in a plastic resin. And then I take a thin slice out of that resin. The resin is basically there to keep the bone from crumbling. Then you glue it to a glass slide, you polish it down, and then it's so thin that light can pass through. You can put it on the microscope, and the picture I have up on the monitor behind me is actually a piece of dinosaur bone. So that's the process, and I've been able to do it and all kinds of cool stuff, including, this is one of my favorites, this is a slice from a T-Rex femur. So I'm histologizing, I guess you could say, is that a new word, this big T-Rex femur, so that's so cool.
I want to get back to T-Rex in a moment, but how do you even grind a slide that's that big?
So for something like this, I actually do have to cut it down into pieces, but I measure the thickness of each cut so that I know how much I'm taking away every time I make a cut, so that I can reconstruct this digitally, and I end up with like a 12 gigabyte file, but it's this T-Rex stitched back together. Sometimes my computers have a lot of trouble with that, but you have to do it.
My colleague, Carrie Levitt-Busion, who also works here, sympathizes with you about that exact problem.
Yep.
Well, since you brought up T-Rex and showed us some amazing slides and samples from it, we actually had a question from a viewer on YouTube who wanted to know if histology could help solve this problem of whether things are juvenile Tyrannosaurus rexes versus maybe another species like Nanotyrannus.
Oh yeah, so histology is one of the ways that we can independently test that hypothesis, and is something that my team actually did last January. We did histological sections of small tyrannosaurs, which, you know, some people say are juveniles and some people say are Nanotyrannus. And we looked at their histology, and we compared the histology to what we see in modern animals. And the histology of these small tyrannosaurs matched more closely with animals that are still growing. They have juvenile tissues compared to what we see in something that's an adult. So our inference from that is that the histology supports that these are juvenile animals. They could be juveniles of something completely different, but right now the only other tyrannosaur we know of in the Hell Creek is T-Rex. So it makes most sense to say it's a T-Rex until someone can show otherwise.
And I love that, because it also shows the process of science that, you know, just like any other scientist, we make hypotheses and then test them, in this case by sampling the histology. Yep. So it's really no different in a way than doing an experiment in a lab.
Yeah, and the cool thing is if the results were opposite and this thing turned out to be an adult, that would also be an exciting result. So testing hypotheses, you're always trying to find evidence against your hypothesis. It sounds wrong, but you want to try to poke as many holes in it as you can. And if it holds water, then it's a strong hypothesis. But, some other evidence one day could come along and, undo the hypothesis that stands right now. And that's how science works. It's progress that way.
Yeah. So I have to ask you about the image in the background there. You mentioned that was actual dinosaur bone under the microscope. And in addition to being really cool science, it's just a beautiful image. Do the colors tell you anything?
Yes. So this is a thin section slide of one of those polar dinosaurs that I was able to section when I was in Australia. And the thin section's under the microscope there, and I'm projecting it to the monitor so everyone can see it. Each one of those colors tells you what direction bone fiber bundles are oriented in respect to the plane of section. So everything that's pink are fibers that are all oriented the same way. The blues and the yellows, all of that just show the fibers are doing different things. And if they're all very orderly, if all the fibers are sort of stacked and regular, that tells you that the growth was slower than if they're crazy colors and going every which way. That shows you that the bone was growing very quickly there.
Oh, that's incredible. There's so much information packed in just a one slide.
Yeah, that's why we should section everything.
Excellent. And there was another wonderful question about bone histology from Dusty on Facebook, who's wondering if you can discover anything about dinosaur diseases.
Oh, yes. So as long as they leave some sort of mark on the bone, then we can learn about dinosaur diseases. And we have found bones that have anomalies to them. These are often broken bones that haven't healed properly. So a lot of times, if you go to any fossil dinosaur exhibit, if you look at the skeleton, sometimes you can actually see these on the ribs. And the ribs, they will have broken, but then they won't have healed correctly. And so you get like this big knot of bone as they're not knitting properly. There's lots of evidence of arthritis in dinosaurs as well, especially in the vertebrae. And then specimens like the famous Sioux T-Rex had huge lesions on its jaw, where you had an infection there that started basically oozing pus, which is disgusting. Whoa. But yeah, so you can learn a lot about dinosaur illnesses and sicknesses by looking at the bone. One of my polar dinosaurs that I studied actually had a horrible injury, and I was able to take a look at its bone tissue. I actually have an example of that here. So let me pull out here. So this teeny tiny little bone is the cross section through a tibia, so the shin bone of a little polar dinosaur. This thin section is the same thing from a different individual. And so in the middle, like right here, that's the only part that looks like this. Everything else is crazy, disorganized bone that shouldn't be there. It's some sort of horrible injury gone wrong, and the bone is just growing everywhere. So this would have been really painful, and this animal did not have a happy rest of his life.
Whoa, that looks like it would be a really hard life. But yep. Clearly survived for a while, at least.
Yeah, right. So it must have been in a lot of pain.
Well, we were scheduled to go for half an hour, but we're getting so many great questions. We were wondering if you're willing to stay on for an extra 5 or 10 minutes.
Yeah, sure. This is fun.
So thank you so much. Thanks for being so generous with your time this evening. So this disease question makes me think of a sort of more general question. You're one of several paleontologists who's part of Polar DinoFest that works at a medical school. And I think not a lot of people think, why are dinosaur paleontologists at a medical school? Could you tell us a bit more about that?
Yeah, that's a great question. And my med students ask me this all the time. So I teach in particular human anatomy to first-year medical students. And they're always very inquisitive about the professors, and a lot of them sort of Google track us and just figure out, because they read like my biography to me, like, oh, I see that you did your PhD at Montana State, and I'm like, yes. So they want to know because it doesn't make sense to them. And what I tell them is, in order for us to understand the past, and it's not just dinosaurs, but anything that's not living today, again, we have to look at the animals that do live today. And, everything has a heart. Everything, vertebrates, tetrapods have hearts. They've got lungs, they've got muscles. So if you can understand how all of this works together in animals that are living today, you can take that idea or those ideas and apply them to the fossil record. And so if you can understand the physiology of birds, then maybe you can have a better understanding of the physiology of their ancestors, the dinosaurs. So when doing this, I got into comparative anatomy dissection as a graduate student, really just studying the different types of vertebrates like cats and dogfish and that sort of thing. And then I was invited to take an advanced human anatomy course, which was just amazing to see those comparisons again, because they're the same muscles in a lot of cases, just configured differently. So from that, it's just a natural progression to, okay, you should be able to teach human anatomy to medical students because you have this baseline biology knowledge. And then on top of that, it's really interesting, like you said, that there's so many places where medical schools employ paleontologists for their anatomy programs. And I tell the med students it's another reason is because we're the ones that think it's really cool and interesting and fun to do. And anyone else that thinks it's interesting and cool and fun to do goes on to be, medical doctors. So if you're there, you're either there to teach it or you're there to learn from it. And we're happy to be here to teach it.
Definitely. And so when you're teaching, you're mostly teaching medical students, but of course, Oklahoma State is a very large university, and you actually have quite a few paleontologists there. Earlier, Zach on YouTube was wondering if there's opportunities for undergraduates or graduate students to get involved in research, paleontology research. Do you have students working with you?
Mm-hmm. So if you go to the website, just type in, OSU CHS, and you'll find it. And go to the vertebrate paleontology webpage there. It tells you about the graduate program that we have. We have both masters and PhDs, degree options. And although the school I work in is a medical school and a graduate school, so this particular branch doesn't accept undergraduates. We don't have an undergraduate program. We do invite undergraduates to come help in the labs. So it's sort of like training and basically a little bit of getting you prepared for grad school. I actually have an undergraduate volunteer that drives 2 hours every week just to help me out in my lab. And it's really helping him decide what he wants to do when he graduates college and it's time to figure out where he wants to go.
That is awesome. Yeah, there are so many opportunities these days now, particularly for undergraduate students to get involved. So seek out those research opportunities where you are. Well, we need to get back to some histology questions because there's so many good ones coming in. Okay. And one of the ones that has been asked by multiple people, both Kim on YouTube as well as Larry from Layton, who I think is one of our museum volunteers, if it's the Larry I'm thinking of. They both wanted to know if you could use histology to determine dinosaur gender.
That depends. That's a great question. And it really depends on whether or not the dinosaur died when it was pregnant. So as you know, birds today are the descendants of dinosaurs. And when female birds are about to lay eggs, they need a bunch of calcium for the shells. And instead of having that calcium being removed from their already super thin hollow bones, they actually start eating a lot of grist, and they actually pack on calcium on the inside of their bone in the marrow cavity, so that when it's time to shell the eggs, that's their calcium reserve and it's not their bones. And so this is called medullary tissue or medullary bone in birds. And only the females have it under natural circumstances. And then once the eggs are shelled and laid, this particular tissue will then get reabsorbed back into the bloodstream. So it's only there in a very brief window of time, but sometimes dinosaurs do die when they're pregnant or after just having laid eggs. And so we do sometimes find this particular tissue fossilized in the bone record. And it's only if we're that lucky that we can say this is probably medullary bone tissue, that's probably a female dinosaur. Otherwise, there hasn't been a lot of good supporting evidence yet that dinosaurs differentiated in size between males and females. There's been a lot of circumstantial evidence. People have been proposing this, but nothing as evident as you see in some mammal species.
And just as a quick aside, here at the Natural History Museum of Utah, we're famous for our collection of the meat-eating dinosaur Allosaurus. And over a dozen leg bones of Allosaurus have been studied histologically, but only one of them showed evidence of possible medullary bones. So it gives a sense of maybe how rare it could be, depending. We have also, connecting to some current or recent dinosaur news, there was this specimen or set of specimens that was in the news lately called the Dueling Dinosaurs that just found a home at a museum in North Carolina. And so a viewer on YouTube wanted to know if you had the chance to histologically sample those specimens, what sort of questions would you ask and what might you cut up?
So that would be amazing to get to do. I think that the museum there has plans to have students there actually do that project, and I'm really excited to find out what they learn. But I would be interested in, from the Tyrannosaur, of course, the leg bones, to see how well that histology matches up with the leg bones of other Tyrannosaurs that I've already sectioned. Again, going back to that differentiating, is it a juvenile versus is it a different species? So if the tissue suggests that it's a still growing individual, then that's a good argument for it being juvenile as well. And the same for the Triceratops specimen that was buried with it. We still really don't know much about how Triceratops grew, because there are not that many articulated Triceratops with, hind legs to go with the giant skulls. So the more we can fill in those gaps, because it's a pretty small individual too, so the more we can fill in those gaps, especially histologically, we could say, this Triceratops was X numbers of years old if we could just look inside its bones. Awesome.
Well, maybe we can, start a campaign to let you sample that specimen.
I'm fine with that. I'll section anything with legs.
Cool. Very cool. So there's just so many great questions. It's hard to choose which one to ask next. But Colin on YouTube was wondering whether you see any differences histologically through time, like comparing late Jurassic dinosaurs to, say, late Cretaceous dinosaurs.
Oh, that's a really great question. It's something that I don't think has been studied in enough detail, but it seems as though the earlier dinosaurs had more what we call plastic growth, where if you sample a large population, this was done in particular with Plateosaurus, a prosauropod. So many individuals were sampled, and some were the same age as others, but they were drastically different in size. And so this is called plasticity and growth, because it's the idea that you can physiologically adjust your growth based on external pressures or stresses. And it seems like there's a lot of plasticity in dinosaurs anyway, but it gets more restricted as you go from, late Triassic into the Cretaceous. There's still some plasticity there, but you don't see the extremes that you do in the earlier dinosaurs. Coelophysis is the same way. There's a lot of plasticity in that early dinosaur.
Do you think that plasticity might have contributed to the early success of dinosaurs?
I think that's definitely possible. And I think it also contributed to the success of dinosaurs throughout their reign, because it makes speciation, I think, a lot easier, because you can, just by evolutionarily adjusting the physiology to stay small, then, you'll selectively breed perhaps with other small individuals and eventually have an offshoot of a different taxon of dinosaurs. So I think this plasticity was probably very advantageous, especially since at this point they were pretty much dispersing across Pangaea and being super successful at pretty much everything.
Very cool. Yeah. And I think, what's really neat is that we see that plasticity not only in the histology, but also in the features of the skeleton as well. So it's cool when two different lines of evidence tell the same story.
Yep. It makes you feel good.
Yeah. As paleontologists, we're used to conflicting evidence. So moving forward, getting back to polar dinosaurs, are there any extinct polar animals, whether they be dinosaurs or other animals, that you'd love to study histologically that you haven't yet?
Oh, wow. Extinct polar animals.
Or living, either.
Well, okay, so I'm a histologist. I love dinosaurs. I love histology. I'm starting to branch out. Don't judge me. I'm getting more into mammal histology now. It just seems to be where some of my questions are going.
That's
so
awesome.
I would really like to histologically sample animals that were dealing with the glaciation and the ice ages, just to see how much of that record could be in the bones, if any at all. There's still a lot to try to figure out about how much you can read out of a bone. Sometimes there could be a structure that's right in front of my face. It could be up on that screen right now, and I don't know what it means, but in 20 years, someone will come along and they'll be like, oh, I can't believe she didn't see this. This means that. And so we need more data, and I would love to section anything for more data and to learn about these crazy times in history where the temperatures were just doing extreme things.
Yeah, it's amazing what we actually don't know about mammal histology. There's so many gaps. Yeah. Mm-hmm. And you pointing out, you mentioned that, we might learn, or a future researcher might learn even more. And so that's why one of the reasons why we keep these histological slides in museums, right, is for those people.
Yep.
Well, I think we'll go with maybe one more question and then start to wrap things up. Thank you so much for staying long and answering all these awesome questions.
I could talk about histology forever.
Totally. So there was a question earlier about the newest dinosaur, but I think I'm going to turn it around and actually ask if there's any sort of current projects that you're working on right now that you're really excited about.
Oh, sure. Yeah. One project that I'm working on, well, there's two actually. So one's with dinosaurs. I'm working on a detailed Tyrannosaurus rex growth curve, incorporating many individuals of many different ages to really try to constrain what T-Rex was doing as it was going from being a little fluff ball up to, mega carnivore. And that project's been taking a while just because T-Rex is so large. I mean, I'm going to have to look at like 8 slides once I finally got this up. So that's been ongoing. But then another project is, like I said, I'm branching out into mammals. I'm trying to learn histologically what the bone tissue of the dire wolf can tell us. And that's made even more exciting recently because this publication just came out saying that dire wolves aren't actually related to gray wolves. They're not like a sister taxon. They're actually a different offshoot of canines. So that makes it even more exciting to me that, I'm already histologically analyzing the dire wolf. I thought I was comparing it to gray wolf histology, and that's what people have been doing. They they look at modern wolves and the pelvis is doing this. It's got to be a 2-year-old, dire wolf. But now we just don't know. Were they doing completely different things?
Well, we'll have to bring you back for Polar Mammal Fest
Yes.
in the future. Yeah, but definitely keep an eye out to hear more about that research as it goes on and gets announced to the public and is published. Well, I want to thank you so much, Holly, for joining us and taking the time to answer all these awesome questions and inspire us with what you do. And a huge thanks to all of our viewers tuning in for this, our third of 6 DinoFest live conversations that are happening this weekend. Our next live stream airs tomorrow at 11 a.m. Mountain Time with more of our amazing Polar DinoFest speakers, Aubrey Roberts, Dan Sepka, and Karen Shin. And we also have additional live streams at 2 p.m. tomorrow and 5 p.m. And remember that this week's live conversations kick off 10 weeks of polar dinosaur exploration with a new dinosaur video from each of our guest paleontologists being released each Friday at 10 a.m. Mountain from now until April 2nd. You can see the complete live stream schedule and watch each Polar DinoFest video when they become available on our website at nhmu.utah.edu/DinoFest. Finally, don't forget that the inspiration for this year's theme, our current special exhibit, Antarctic Dinosaurs, is open at NHMU through April 4th. Be sure to book your tickets online in advance for your Polar DinoFest experience, where you can have an in-person visit with the exhibit. Polar DinoFest has been made possible by the Alternative Visions Funds and Utah Legislature and Utah Division of Arts and Museums. Many thanks to these sponsors for helping bring Polar DinoFest online. And finally, thank you again for tuning in, and we hope you join us for our next panel tomorrow at 11 a.m. Mountain Time. And remember that if you missed any of the previous live streams, they've been recorded and will be available on our website, YouTube channel, and Facebook page. Thank you once again, Dr. Woodward Ballard, and have a great evening.
You're welcome. Thanks for inviting me.
Every year, NHMU's DinoFest brings you face-to-face with the world's top paleontologists and their incredible discoveries. Now in its 5th year, DinoFest is going online with 10 weeks of virtual exploration of the frozen world of polar dinosaurs. From the Alaskan North Slope to Antarctica, dinosaurs are found on all 7 continents. But how did they survive at the extreme ends of the Earth? Join us online each week starting January 23rd as we introduce a different paleontologist whose research has helped uncover a thriving polar world far different than what we know today. Polar DinoFest starts online January 23rd and continues through the run of our special exhibit, Antarctic Dinosaurs, open through April 4th. Don't miss it.